Hi. All right, next question is from Barb. Good morning again, Barb. Yeah, good to see you're still here. Let's see. Hi, Dr. Ben. Is there a point at which the virus may mutate itself out of existence? I think with Ebola, it was so virulent that it would kill us before it could spread the way COVID-19 does. Yeah, we like to think that sort of stuff. It might be true, and yeah, we're still here, so. Yeah. Ebola was not a good thing for us, but that wasn't good for the virus either. Yeah, that's the idea. But Ebola isn't a human virus, of course. It probably persists for life in its natural hosts, which are the bats. So it's just some of them that spill over into humans and then go extinct and then whatever. It just keeps happening, yeah, over and over. But it doesn't do anything particularly good for the long term benefit of Ebola virus. So, yeah. All right, good question. This has actually been addressed experimentally in a couple of different ways. So, um, what would happen if, yeah, if a virus just started making mistakes and started making too many mistakes? So we have old experiments from somebody called Shane Crody, and what he was looking at is like how many mistakes can you have in a virus before the virus just, on average, is dead. And he figured this out for poliovirus, and if I remember right, the number, like the magic number, the threshold was somewhere around 4 or 5. So you could have, let's say you could have 4 mutations and you'd be fine, and then once you get to 5 mutations, then you're probably dead. But even at 3 mutations, you've got some viruses that are dead. And at 4, there may be 50-50, and at 5, you've only got maybe 10% that make it. 6, 7, 8 mutations, and yeah, your virus is just almost always dead. Because mutations are generally bad for you, bad for the virus, bad, yeah. Bad mutations, yeah. Um, but the coronavirus also has a protein called NSP14, and that's a great name for a protein, isn't it? It is an exonuclease, and an exonuclease will cut away nucleotides that are misincorporated. That's what we think. So the virus is reading along, and it's following its little template, putting in everything right, and then oops, it made a mistake right here. What does it do? We think it calls in NSP14 that then cuts out that mistake, and then it can continue to go on. And it turns out NSP14 has a bunch of other functions, that are all way too esoteric to go into here, but that are related to the way that the virus expresses its genes. Yeah, whatever. You can go look it up if you want, or somebody ask and I can tell you. Um, and so the question was, what happens if you knock out the active part of that? So you leave the whole protein there in case it's sticking to something else and the other thing won't work if you just totally delete this part. That's a pretty, yeah, scorched earth way to do your reverse genetics study, just deleting things, yeah. And what they found was that you could do this to the virus. And at first, it suffered a little bit, you could tell, and it just started building up mutations. But it built up these mutations, and it was actually all right. They thought that the virus with these mutations would probably just go extinct and would be, outcompeted by other viruses almost immediately. And it just didn't happen. The virus didn't drive itself to extinction. It ended up being in a stable, highly mutagenic state, which is kind of neat. Yeah. So what's going on there? We know viruses make a lot of new viruses, new copies of the virus. So we think it's on the order of one virus goes in and about 10,000 come out of each cell. So 10,000 times multiplication for every single cell you infect, and then the virus will then infect lots of cells. If it infects 10,000 cells, then boom, multiply that by 10,000, and you start to get numbers, yeah, that are very big and also very true. So it looks as though the virus, yeah, was putting in all these mutations, but most of these copies of the virus were probably dead, and I think they might have even looked at this in the paper and shown that far fewer than normal of the individual virus particles were actually able to start an infection. But some of them were. There were those few out there on the edge, and those were the ones that then went on to the next round. And so they still had a broken proofreading mechanism, but they also had all these mutations, but they also had survived somehow. And so those then go on, and they found another, generation of viruses, which have all the mutations that the parents started with, plus all the brand new mutations that they put in. And they just kept passaging it, kept growing the virus, and sure enough, it kept going. So yeah, you can make a hyper-mutating version of a coronavirus, and it doesn't necessarily kill off the coronavirus. Doesn't necessarily make it any stronger. These experiments probably only work in cell culture, and cell culture is like a spa day for a virus. Because the virus, it just wants to get into a cell and relax and unroll its genome and start making proteins. That's what the virus is there for. And you prepare this beautiful little layer of fully healthy, very nice cells that are the favorite kind of cells for your virus to grow in. And you put in the most lovely growth medium on there. It's like Powerade, but with extra sugar and amino acids in it. And you put the virus in there. There's no competition. There's no immune system. Quite often, the cells have actually been disabled, so they can't even recognize that a virus is there and make any kind of response. And the virus just grows. And so, in this situation, you can get a virus that is super mutated and is still able to function in this situation. Question is, if you threw that back into an actual mouse, what would happen? I don't remember them doing that in this particular study, and that would probably be worrying, I would think. Yeah, because they would say, well, you've hyper-mutated the virus, and now you want to go put it back into animals, and, what if it is bad or something? Which probably not, but yeah, you'd have to do that experiment. And so we don't know that they could survive in the world. I would lean toward no. Competition and nature are a pretty strong selective force, and they're real good at weeding out the weak, and so that's why the coronavirus doesn't change more than it already does. I think that's the best explanation. So that was very long and winding, but I feel like we got there. So in practice, in a human body, yes, you can probably drive a virus extinct by forcing it to mutate itself out of existence. And there are some drugs that are aimed at doing exactly that. But in cell culture, if you want to just, test out a thing, no, you can make a coronavirus that just keeps on mutating, and it just keeps getting weirder. And so far, there's no particular end in sight. It's really fascinating. So thanks very much. This has been Ask Dr. Ben.